icy landscapes, fungi-infested crops, and nanowire networks? Today, we're diving into the latest from the world of scientific research. Welcome to the Science Hangout. Researchers from the University of California, Irvine, and NASA's Jet Propulsion Laboratory have discovered that the impact of polar ice deterioration on future sea level rise may have been greatly underestimated. By analyzing satellite radar data, they found that the grounding line of Greenland's Petermann Glacier shifts significantly during tidal cycles, allowing warm seawater to intrude and accelerate ice melt. This behavior, not previously considered in climate models, makes glaciers more sensitive to ocean warming. Including these dynamics could increase projections of sea level rise by up to 200% for not only Petermann Glacier, but also other glaciers in Greenland and Antarctica. The study emphasizes the need to incorporate ice-ocean interactions in climate models to improve predictions of sea level rise. Scientists warn that fungal diseases in crops pose a devastating threat to the global food supply. Despite the use of antifungals, growers lose 10 to 23% of their crops annually to fungal infection, with additional losses after harvest. Global warming is causing the spread of fungal infections, potentially affecting more countries. Fungi's tolerance to higher temperatures increases the risk of infecting animals and humans. Fungal infections could lead to a global health catastrophe and food shortages impacting billions of people. However, new antifungal chemistry shows promise in targeting multiple mechanisms and reducing resistance. Urgent action, investment, and a unified approach involving stakeholders are crucial to protecting crops and ensure food security. Researchers at the University of Sydney and Japan have found that nanowire networks can exhibit both short and long-term memory, resembling the human brain. The networks, made of highly conductive silver wires in a mesh-like structure, mimic the brain's physical architecture. The researchers conducted memory tests on the nanowire networks, which showed cognitive function similar to human psychology experiments. The networks could remember a desired endpoint in an electric circuit, similar to the end-back task. By manipulating voltages, the researchers guided the pathways of the nanowire networks and achieved stable memory accuracy over time. The study also demonstrated information consolidation and storage in the nanowire network, akin to short and long-term memory in the human brain. This research holds promise for advancements in artificial intelligence and neuromorphic computing. Researchers at Duke University have made a trailblazing discovery regarding the existence of electric fields within and around biological condensates, which are cellular structures that form compartments without the need for a membrane. Similar to the imbalanced electrical charges found between microdroplets of water and air, these electrical fields within condensates could spark reactive oxygen reactions. The findings, published in the journal Chem, have implications for understanding biological chemistry and the energy sources that may have contributed to the origin of life on Earth. The researchers suggest that the phenomenon could explain how reaction energy was obtained in a prebiotic environment. The study sheds light on the importance of interfaces in biological systems and the potential chemical functions of condensates. A study led by researchers at Yale and the Broad Institute of MIT and Harvard reveals that what is missing in the human genome compared to other primates may have played a crucial role in human development. Published in the journal Science, the study explores the loss of approximately 10,000 genetic elements over the course of human evolution. These missing genetic pieces, some of which are associated with neuronal and cognitive functions, differentiate humans from their closest primate relative, chimpanzees. The researchers found that these genetic deletions, present in other mammals, are common to all humans, suggesting their evolutionary significance. The study highlights that not only adding, but also removing genetic code can have profound consequences for human traits. The research utilized massively parallel reporter assays, MPRA, to examine the function of thousands of genetic changes among species. The findings contribute to a deeper understanding of the human genome and the factors that make humans unique as a species. DNA data storage is no longer a distant concept, but a near-future possibility, with the first DNA data center expected to be operational within the next 5 to 10 years, according to Professor Tom de Greef of Eindhoven University of Technology. Instead of storing data as zeros and ones on hard drives, data would be stored in the base pairs of DNA, AT and CG. Synthetic DNA would be used, with bases synthesized and arranged to form DNA strands that encode files and photos. although the technique is currently suited for archival storage due to expensive data retrieval, DNA storage offers advantages such as compactness, longer data lifespan, and the potential to render large energy-consuming data centers obsolete. The concept of using DNA strands for data storage emerged in the 1980s, but remained impractical due to its complexity and high cost. However, advancements in DNA synthesis over the past 3 decades have at last made it technologically feasible. Currently, the most significant challenge is the reading of stored data in DNA. Presently, the PCR method used for reading, known as random access, consistently causes errors. The biggest problem being that data quality deteriorates with each read. These factors have stopped DNA data storage from becoming widespread. Professor Tom DeGreef, together with PhD student Bas Bogels, Microsoft, and a group of university partners, have developed a scalable technique for DNA data storage, addressing challenges related to data reading. The technique involves encapsulating individual files in microcapsules made using proteins and polymers, each containing a single file. The capsules contain thermal properties that can be manipulated. By raising the temperature to above 50 degrees Celsius, the capsules seal themselves, which allow for separate PCR processes to happen inside each capsule simultaneously. Dubbed thermo-confined PCR, this innovative approach reduces errors significantly. At the time of the article being published in the journal Nature Nanotechnology, the researchers had successfully managed to read 25 files simultaneously with minimal errors using their novel method. By lowering the temperature, copies detach from the microcapsules and the integrity of the original files remains largely untouched, with only a 0.3% quality loss after 3 reads, while the current existing methods for DNA data reading leads to a 35% file quality loss per reading. Moreover, by incorporating fluorescent labels and color-coded capsules, the data library becomes easily searchable. A device recognizes the colors and easily separates the capsules. It's not hard to imagine a robotic arm neatly selecting requested files from a pool of capsules in a DNA data center. It's very cool to think that we could have the world's first DNA data center very soon. Researchers have developed a new technique to overcome a major hurdle in treating the deadly brain cancer glioblastoma. Currently, the most potent chemotherapy drugs can't reach the aggressive brain tumor due to the blood-brain barrier. But now researchers have used a skull-implantable ultrasound device to open the barrier and deliver chemotherapy directly to critical regions of the brain. The procedure only takes 4 minutes and the patients can be awake during it. The treatment has been proven to be safe and well tolerated, with some patients receiving up to 6 cycles of treatment. After a few hours, they can even go home. Using ultrasound technology to open the blood-brain barrier, the researchers observed a significant increase in drug concentrations in the brain, around 4 to 6 times higher than usual. This breakthrough allowed powerful chemotherapy drugs like paclitaxel and carboplatin to reach the brain. Additionally, the study revealed that the blood-brain barrier recloses within 1 hour after the procedure. This critical finding will help optimize the delivery of drugs and maximize their penetration into the brain. Lead researcher Dr. Adam Sonnabend from Northwestern University Feinberg School of Medicine called this advancement a potential game changer for glioblastoma patients. The current chemotherapy drug used for glioblastoma, called temozolomide, is weak compared to the potent drugs that can now be delivered to the brain using this technique. The scientists are also conducting an ongoing phase 2 clinical trial for patients with recurrent glioblastoma. In this trial, patients receive a combination of paclitaxel and carboplatin delivered to their brain using the ultrasound technique. The objective is to determine whether this treatment can extend the patient's survival. The incredible work of these researchers in this study not only opens new possibilities for treating brain cancer, but also paves the way for novel drug-based treatments for millions of patients suffering from various brain diseases. It's an incredibly exciting development that will definitely bring hope to many.